I am Snigdha Bharadwaj from ITS College of Pharmacy. I welcome you all in this session. In this session, we will be continuing our last topic of general production of enzymes, general consideration part 2. So, let us get started. So, we have discussed amylases and catalysis in the earlier session. In this, we have some other enzymes to be discussed as per the syllabus of AKtU. So, third enzyme is proteases. So, proteases is a mixture of peptidases and proteineases. Are these enzymes that performs the hydrolysis of peptide bonds. So, basically proteases what it is meant for, it hydrolyzes the peptide bonds present in enzymes or present in any compound. So, we use these type of enzymes proteases for this purpose. Peptide bond links the amino acid to give the final structure of protein. So, basically the peptide bonds are linked the amino acids to give the final structure of a protein. Proteineases are extracellular and peptidases are endocellular. As we have discussed in last slide that the enzymes present within the cytoplasmic material are considered to be as endocellular within the cell, and those who are going outside from the cells or penetrating outside from the cells during any fermentation process are considered to be exocellular. So, proteineases are extracellular or exocellular and peptidases are endocellular type of enzymes, because proteases is a group of enzymes. Second most important enzyme produced on a large scale after amylases. So, it is considered to be a second most important enzyme for industrial purposes. Now, based on its categorization, it is of two types, like based upon residue in the catalytic site and based upon pH in which proteases are active. So, based on catalysis site and pH. So, these are the two broad classification of proteases. So, based on catalytic site, serine proteases, threonine proteases, aspartame tate proteases, cysteine proteases, glutamic acid proteases and metalloproteases. These all are catalytic site based proteases which targets different amino acids, right. And based on pH, we have alkaline serine proteases, acid proteases and neutral proteases. So, we have a group of proteases based on pH, those who are active on neutral pH, those who are active on acidic pH and those who are active on alkaline pH. So, this is the broad classification of proteases. Proteases, now why it is important to produce proteases, or what is the role? As we know that it hydrolyzes the peptide bonds. So, we can use it in detergent industries, like it is stable at high temperature, it is stable in alkaline range between 9 to 11, right. Stability in association with chelating agent and perborates, right. So, it is also stable in complex formation with chelating agents and perborates, but the shelf life is affected in the presence of surface active agent. But if we add surface active agents or surfactants to the product which contains the proteases, so the shelf life might get affected. Now, we will discuss about alkaline serine proteases. pH of the production medium is kept at 7 for satisfactory results. Although it is alkaline serine proteases, but if the pH lies at 7 point or nearby 7.0 range pH range, then the enzyme gives satisfactory results, okay, right, during production or fermentation of these type of enzymes. Have serine at the active site alkaline serine protease. So, serine is an amino acid which is present in these type of particular enzymes and at the active sites. Optimum temperature maintained is 30 to 40 degree centigrade. So, the optimum temperature is here, which needs to be kept for during fermentation process is 30 to 40. Important producers are B. licheniformis, B. amyloliquefaciens, B. firmis, B. megatherium, Streptomyces griseus, Streptomyces freddi, Streptomyces rectus, and fungi also. So, variety of microbes can be used for the production of proteases, different kind of proteases, right. So, here are some examples. So, fungi like Aspergillus niger, Aspergillus oryzae, Aspergillus flavus are the fungus which can be used to produce proteases of alkaline serine type, right. Enzyme used in detergents are chiefly proteases from Bacillus strains. So, those who are getting used in detergent industry are basically isolated or produced from proteases from Bacillus strains, specifically Bacillopeptidases. So, these are the just basic information regarding the enzymes which is getting produced. So, best known proteases are Subtilisin, Carlsberg. This is also known as, you can say, a recognized name of the enzyme from its production source, right. So, this proteases known as Subtilism Carlsberg is produced from B. licheniformis and Subtilism novo from B. amyloliquefaciens. So, these are highly used we can say proteases which can be procured or produced from these type of species or microbial or bacterial species. These enzymes are not inhibited by EDTA, ethylene diamine tetraacetic acid, but are inhibited by DFP, diisopropyl fluorophosphate, right. So, these are its chemical properties. During the production we need to consider all the cofactors when as we discussed in the last lecture, that cofactors also affect enzyme functionality or behavior during the production because of these compounds. So, we should know and we must know which type of cofactor is affecting the enzyme positively and negatively, right. Likewise, to prepare a suitable encapsulated product, a wet paste of enzyme is melted at 50 to 70 degree centigrade. So, if we want to encapsulate these enzymes, so the produced enzyme mass needs to be melted at 50 to 70 degree centigrade with a hydrophobic substance such as polyethylene glycol and then converted into tiny particles. So, here it comes the concept of microencapsulation. We have read in NDDS or industrial pharmacy. So, there you can see how the microencapsulation done using polymeric materials or different types of polymers. So, in this case we just converting our isolated enzymes and coated with some polymeric material to make tiny particles. So, fermentation, how it is produced. So, here it is a flow chart. So, culture are stored in the hydrophilized state, lyophilized state or under liquid nitrogen, right. So, cultures, lyophilized means lyophilization or freeze drying, right. So, it is also the terminology also comes from pharmaceutical engineering, lyophilization or freeze drying. So, lyophilized culture or under liquid nitrogen. So, culture means nutrient medium in which we put the inoculum, we put our bacterial strain to grow, right. So, cultures are stored in the lyophilized state or under liquid nitrogen. Initial culture are carried out in a shaken flask and small fermenters, right, like 4 to 100 meter cube at 30 to 37 degree centigrade. So, first the culture are stored in lyophilized state or freeze drying state or under liquid nitrogen, and then initial cultures are carried out into shaken flask method using small fermenters at 30 to 37 degree centigrade. Fed batch culture is generally used to keep down the concentration of ammonium ions. So, in last slide we have also discussed the type of fermentation process, solid state, submerged fermentation. So, in here a fed batch fermentation or culture is generally used to keep down the concentration of ammonium ions and amino acid as they may repress the protease production. So, this is important consideration here, that why we use here fed batch type culture, because during the production or fermentation of these proteases, so it might due to the concentration of ammonium ion and amino acid, it may affect or repress or suppress the growth of enzyme we are producing. So, ultimately it will affect the yield of enzyme we are producing. So, high oxygen partial pressure is generally necessary for optimal protease tires. So, here these are the cofactor, high oxygen and partial pressure. Then you need to maintain the level of oxygen quite high, and partial pressure is normal, is generally necessary requirement for the production of proteases to get higher yield. Time span for fermentation is 48 to 72 hours depending upon the organism, which type of bacterial strain or fungal strain you are using. So, accordingly you will variate the range between 48 to 72 hours for the production of particular enzyme. Proteases must be converted in a particulate form before they are added to the detergents. Yes, of course, in last slide we have discussed that tiny particles or micro encapsulation we can say, right. So, it must be converted into micro encapsules, the tiny particles, before final use. So, here it is written like this only. So, this is the basic fermentation chart, although the steps can be varied for depending on different type of species you are using. So, but this is the basic flow chart. Now, we will talk about after alkaline serine protease for neutral proteases, right. They are relatively unstable and calcium, sodium and chloride must be added for maximal stability. So, the most important type of proteases are alkaline serine proteases because of its higher stability in any type of environment, and then neutral proteases comparatively are unstable and calcium, sodium and chloride like ions must be added for maximal stability for improving stability of these neutral proteases. No stable at higher temperature, they are not preferable for higher temperature usage. Producing organisms are Bacillus subtilis and Bacillus negatorium, etc. So, Bacillus type of bacterium species are usually taken for producing neutral proteases. They are quickly inactivated by alkaline proteases. So, this is the disadvantages of neutral proteases, right. Acid proteases, similar to mammalian pepsin, it is quite similar to mammalian pepsin. It consists of rennin-like proteases from fungi, which are chiefly used in cheese production. Okay. So, basic application of acid proteases is in cheese production. They are used in medicine, in the digestion of soy protein and for soya sauce production, and to break down wheat gluten in the baking industry. So, the basically acid proteases main uses in food industry and more specifically in bakeries, right. So, here are the significance of developing these enzymes, why we are doing fermentation and we are producing these enzymes at large scale, because they have commercial applicability. Proteases applications. So, textile industry to remove proteinaceous sizing. In textile industry also proteases type are used. Silk industry to liberate silk fibers from naturally occurring proteinaceous material, like from the organism or from the insect, we separate out silk fiber using these type of enzymes. Tendering of meat, yes, in food industry and used in detergent and also food industries again. Next is lipases. Lipases are also called as glycerol ester hydrolysis. They are subclass of esterases. It splits far into mono or diglycerides and fatty acid. Lipases breakdown of lipids, right. So, it split fast. Also we can say it breakdowns into mono or diglycerides or fatty acids, right. They are extracellular enzymes mainly produced by fungi. So, these are mainly produced by fungi like Aspergillus, Mucor, Rhizopus, Penicillium, etc. Bacteria producing lipases include species of Pseudomonas, Acrobacter and Staphylococcus. So, these are basically gram negative bacteria which produces lipases. Yeast like Torulopsis and Candida are also commercially used. So, yeast or molds like Candida and Torulopsis are also used for producing these type of lipases. So, mode of action basically, as it says that it breakdowns fats into mono and diglycerides and fatty acids. So, here we have triglycerides, lipases in presence of water. So, it breaks down these triglycerides into di and again mono. So, fatty acid and fatty acid. So, here it is work like that. In the presence of lipases, triglycerides will get converted or break down into di, and again on simultaneous reaction we can get monoglycerides and fatty acids. Factor affecting. So, enzyme production must be induced by adding oils and fats. So, factor affecting means during production we need to consider certain cofactors and associated factors, so that we can have the better yield without affecting the enzyme performance. So, enzyme production must be induced by adding oil and fats. During production of lipases we use oil and fats for its production, but in some cases the fats have effect on lipase production, but in some production, as we have variety of microbes here, we can produce lipase from fungal source, we can produce from bacterial, and we can produce from yeast. So, all these types must be having some sort of variation during production. So, here it is written some species can induce the growth using if we use fat and oils, but some cannot, right. Glycerol, a product of lipases action, inhibits lipase formation. So, during fermentation we need to understand if we add glycerol, and so it may affect lipase formation. So, we can get the slightly less yield of that particular enzyme. Lipases are generally bound to the cell and hence inhibit an overproduction, but addition of a cation such as magnesium ion liberates the lipases and leads to a higher enzyme titer in the production medium. That means, lipases generally bounds to cell. So, it inhibits the overproduction, but if we add cation to the fermentation medium or culture, so we can expect the high yield of this particular enzyme. What are the different applications? Primarily marketed for therapeutic purposes as digestive enzyme to supplement pancreatic lipases. So, commercially for the treatment of pancreasis disorders, it can be administered. Since free fatty acid affect the odor and taste of cheese, and the cheese ripening process is affected by lipases. Microbial effects during the aging process can be due to lipase action. So, in food industry also it can be used for cheese ripening during the which adversely affect the cheese ripening or odor and producing bad odor during the process. So, it can be used in that. In the soap industry, lipases from Candida is used to hydrolyze oils. So, these are some few applications of lipases. Next is peroxidase. So, it is a hemoprotein catalyzing the oxidation of a number of substrate by hydrogen peroxide. Basically, it is a hemoprotein which is which catalyzing the oxidation, affect the oxidation of a number of substrate by hydrogen peroxidases. Peroxides found in wide variety of organisms from plant to human to bacteria. Basically, its function to break down hydrogen peroxide, which is one of the toxin produced as byproduct of using oxygen for respiration. So, Bacillus and Aspergillus species produces the peroxidase enzyme, which has the potential to degrade various hazardous compound. Basically, it is targeted here. Hydrogen peroxide is considered to be a hazardous compound. So, it is used for breakdown for breaking down that hydrogen peroxide. Examples are E. coli, Streptomyces species. These are the some examples through which we can produce peroxidases. Mycobacterium tuberculosis, Klebsiella pneumonia, Rhodobacter capsulatus, Stereotriticea, etc. Applications are for the treatment of industrial waste, it is breaking down hydrogen peroxide. Phenols, which are important pollutants, can be removed by enzyme catalyzed polymerization process using horseradish peroxidase. It can also be converted toxic material into less harmful substances. So, basically it is useful for waste management processes. In many manufacturing processes like adhesive, computer chips, part and lining of drums, examples polymerization of anilines and phenols in organic solvents matrices. As a histological marker like cytochrome C peroxidase is used as a soluble, easily purified model for cytochrome C oxidase. Okay. So, for histological analysis we can use it. Now, the last is penicillinase. So, penicillinase is responsible for inactivating penicillin and cephalosporin. The penicillinases are of two types. Penicillinase basically inactivate penicillins. Penicillin acylase or amidase produced by Aspergillus penicillin, penicillium or mucor. These acts on amide bond present in penicillin, through which the acyl group is attached to the basic nucleus, or that is 6 amino penicillinic acid. So, basically it targets this nucleus and break down its chain through acyl group. Lactamases, this is another type produced by Streptococcus species, which attack the lactam ring of penicillin and give penicillinic acid. So, here you can see penicillin in presence of penicillinase, hydrolysis. So, this is penicillin, which penicillinic acid, through which we can get this particular thing. This ring is now affected due to presence of penicillinase. Source of penicillinase is Bacillus species and also certain strains of Staphylococcus. Responsible for causing overwhelmingly penicillin resistance, the cause of an infection. During penicillin resistance we can use it. Contamination in penicillin fermentation medium, aids in the rapid degradation of penicillin in the penicillin fermentation medium, right. Those fermentation processes which are getting affected through the presence of penicillin, we can use penicillinase to break down the to deactivate penicillin effect. Penicillinase obtained from B subtilis and B cereus represents the industrially produced enzyme, which exert action to some extent in the removal of penicillin via specific inactivation. When manufacturing the finished preparation, penicillinase is isolated from the culture liquid, sterilized, lyophilized, and the finished product is supplied in glass vial, which are placed in carton boxes. So, applications to control of sterility and microbial purity of beta lactam antibiotic substances, control of sterility, beta lactam antibiotic pharmaceutical products, microbiological control during manufacturing of these type of antibiotics, quality control of beta lactam antibiotics in control, analytical laboratories during their certification. So, these are some references. I hope you understand the significance and industrial application of all these enzymes very well, and I hope you like my presentation. Thank you and stay safe.